Hello, my name is Dr. Stacy Zuber. I am the Illinois State Soil Health Specialist with USDA NRCS. And in this session, I'm going to be talking to you about soil biology, the underground workforce. So we're going to go through and answer a number of different questions about soil biology. Um, and you may have heard before in other presentations and other people in other conversations about how important soil biology is to soil health. So we're going to kind of dive into some of those details. And we're going to start with, what are they? What are we talking about when we're talking about soil biology? How many of them are there? How abundant are the soil organisms within your fields, within different acres? What do they do? What are their skills? What can they bring to the table? And then what can they do for you? What can they do for your soils, for your field, for your farm, for agronomic decisions that you make? We want to talk about where soil biology can fit into all of those things. And then finally, how can you recruit that soil biology? How can you get them in your soils? And then how do you keep them there? So when we're talking about soil biology, there's a lot of different organisms. So we're going to start in this. This is a soil food web. It's a simplified version of all of the energy flows that are happening within our soil among all these different organisms. So we have over here on the left side, we have the sun and plants and organic matter. And then we get into some of our very small organisms in the soil, our microorganisms, and that's going to be our bacteria, our fungi, protists, also called protozoa. So we have these very small organisms, and then we go out from there. That energy keeps flowing out to these other larger organisms, our nematodes. We'll talk some about nematodes. I know a lot of the times people think of nematodes as bad because we have soybean cyst nematode. They're not that is a problem, is a pest for us, can cause a lot of problems, but they're not all bad. In fact, most of them are very beneficial, and we're going to talk about why and how that may be, and why you do want a good amount of nematodes within your soil. Earthworms, and then we can go out from there into the arthropods, our beetles or spiders, and then out from there into mammals and birds. So we're mostly in this presentation going to be focused on this section of the food web. We're going to be talking about some of the very small, and then the next larger organisms is we're going to focus on in this presentation. So this is just several different pictures of the different organisms within our soils. So this is kind of showing the diversity of life that we have in the soils. So everything from over here, we have mycorrhizal fungi, we have nematodes, tardigrades, earthworms, all sorts of different organisms. And this is just a sampling of them that's in there. I just wanted to share with you some of these pictures of all the different types of organisms that we have within our soil. So how many of them are there? Well, in 1 teaspoon of soil, like we see here over on the right, we have 1 billion, up to 1 billion bacteria, up to 1 million fungi, and up to 100,000 protozoa. So those are some very large numbers that we're finding in just that small 1 teaspoon of soil, but that's not all of them. There's more in there. There's yeast, there are algae, there's microscopic insects, there's all sorts of other organisms that are also in that 1 teaspoon of soil. So ultimately, if you add them all up, there are more microorganisms within that single teaspoon of soil that you see there on the right than there are the number of people on this planet, which is a very large number of organisms in a relatively small amount of soil. So that can kind of tell you in your fields, when you have an acre or 10 acres or 20 acres or 100 acres, how many of these organisms you have out there already. All right, so here's just looking again, another way of looking at this, where we have the numbers of these different types of organisms in a cubic meter of soil, which is equivalent to about 1.3 cubic yards of soil. And we have on here, bacteria, fungi, algae. These are our smaller ones. Protozoa would also fit in here as well, the smallest of these organisms. And you can see how high the numbers, the abundance of these organisms are. And as you go to the right, these tend to get larger and larger organisms from left to right, and the numbers are much smaller, which is the pattern we see across all sorts of ecosystems. Is as organisms get larger, their number gets smaller. And so we see that all over. But I just wanted to show you here kind of how we have this distribution in number for the different organisms that we have. But another takeaway from this is that it kind of lets us see how much weight there is in those organisms. So we've talked about the number of organisms, but now if we talk about the weight of those organisms, we have in 1 acre of healthy soil, that's going to contain almost 7 tons. So 6.7 tons of living organisms is in that single acre of healthy soil. And that's about equivalent to 8 cows. All right. So a huge amount of biomass in those organisms living in our soil, and the number of them is very high as well. So just if you take nothing else away from this, just the number of organisms that you have in that soil in your fields is a lot. There's a lot of them out there. So what do they do? What are their skills? This right here, that the fact that soil biology, all these organisms that are out there in those soils are responsible for the functions of that soil. That right there is why we talk about soil biology being so important for soil health. And that's because when we're talking about soil health, we're primarily talking about how well that soil functions. What is its capability of performing soil functions? So it's the soil biology that's responsible for most of that. So if you have a large enough soil microbial community, if you have a diverse enough soil community, microbial community, biological community, then you're more able to do those functions, perform those functions well and efficiently, and be able to have good functioning soil, which means your soil health is higher. So when we're talking about what they do, we can categorize them by what they do. What functions are they able to perform? And so we have 3 main groups of function that we can group these by. They're functional groups. And so we have several different types of organisms that fit into each of those functional groups. And there's some types of organisms that can fit into multiple functional groups because they do multiple things within that one type of organism. So we're going to start with the ecosystem engineers. So these are going to be doing decomposition, carbon cycling. They're going to be responsible for building soil, creating aggregates, and pore spaces. And then those two together are really important both for ecosystem resiliency. So when we're talking about ecosystem engineers, we're talking about plant roots. So we're not just talking about, the insects and worms and things like that. We're also talking about plant roots that are in there are going to be really important for this, for creating biopores and creating spaces within the soil and helping to contribute to aggregate formation. We have earthworms that are in there. We have larger invertebrates like millipedes and beetles. And we have some pictures down there at the bottom of this slide showing some of those different types of organisms in there that are doing that are responsible for some of the decomposition of organic matter, of residues on the soil surface, and then also building those pores and helping to develop the soil structure. So our next functional group is the biological regulators. So again, they also contribute to decomposition and carbon cycling, but when we're talking about them, a good amount of what they are contributing to is population regulation. So they are controlling the other populations, the other organisms, how large those populations are, how abundant they are within the soil. And then as part of that, they're also contributing to nutrient cycling. So we'll talk about this a little more, but it's really a predator-prey relationship that we're talking about with these type of organisms. And they are the predators that are eating other organisms. And by consuming those other organisms, they are able to control their population. And so that is also all of that is really closely related, again, to that ecosystem resiliency and the plant productivity as well. And so the organisms that we have in there, this is where those nematodes come in. We have protozoa. There's some other small invertebrates that are in here as well. But all of those are really important for being predators of other organisms and controlling this population. So actually in some of these, we have some really interesting pictures here. This is a picture of an amoeba, which is a type of protozoa. And in here, we can see all of these green parts in this picture are bacteria that are being consumed, ingested by this amoeba, by this protozoa. And then we have nematodes, mites, and then here, this is actually a really interesting picture where we have a fungus that's actually preying on a nematode. So the nematode is the prey of this fungi. And what it does is it actually creates a trap for the fungus, for the nematode that goes through this loop. And then the fungus tightens down on it and is able to kill it and consume it. So there's an instance here where we have a fungus, which is actually the predator of a nematode. And then finally, our third functional group is chemical processors. And so again, all three of these are really closely related to decomposition with carbon cycling. Chemical processors are also related to a number of these other ones. So they're also contributing to building soil, creating aggregates, not as much the pores. That's the larger organisms that are contributing to that. But the aggregates is really where we see a contribution from the chemical processors. We see nutrient cycling, really important for that. And then a little more of a direct contribution to plant productivity. All right, so the organisms that we're talking about as chemical processors, those are basically our soil microbes, our bacteria, fungi, protozoa. And so here we have some pictures of those. We have some bacteria, different types of bacteria. We have fungi, we have a protozoa. But one of the key things with this is 90% of all of the energy in the soil is flowing through this functional group, through these organisms. Right, so this is just putting it all together where we have all of these three different groups. And I just want to point out, ecosystem resiliency and plant productivity, ultimately, those are kind of our two functions of this, like that we are really, really, really interested in because they help us from in agriculture. We need these two functions really working. We need productive plants. We need a resilient system. And so all of these different functional groups are contributing to those two functions, either directly or indirectly. We see that where there's that contribution from the soil biology. So let's talk a little more about how they're doing that. Are they contributing to that resiliency, that plant productivity, and what can they do for you? So again, remember, they are the drivers of soil functions. So how can the soil biology improve those functions for you, for how we need to use them in agriculture? So first of all, through nutrient availability, trafficability, water infiltration, resilience to stress. So that's, goes back to that resiliency, and then disease suppressiveness. So we're going to go through a little bit on each of these and how the soil biology can kind of improve those. So we're going to start with nutrient availability. And this is just a very simplified version of the nitrogen cycle. And it's showing simply the parts of the nitrogen cycle that are regulated entirely by some sort of biological organism. So here we have the nitrogen fixing bacteria through here as well. And then we have nitrifying and denitrifying bacteria that are doing conversions of different inorganic nitrogen forms. And then another really important part is these decomposers. Our bacteria, our fungi, some of the larger organisms are what as well would probably be in here, where they are contributing to the decomposition of plant residue. So we see this just obviously involved in the cycling of nutrients here. And then as they're cycling those, the plant available forms here can become more available to your plants, to your crops. All right, another way that we can see this relationship between the microbes, between the soil biology and nutrient availability is actually through a process that's called priming. And so it's kind of like if you think about priming the pump, you're getting it started, you're getting it going, you're getting it flowing. We see that here. And I'm going to go through the steps of how this works. But basically, it starts with the plant roots are attracting the microbes. How they do this is through exudates, root exudates, which are basically carbohydrates and proteins, and they're secreted by the roots. And so they are right in the area around those roots. And basically, that is a food source for the bacteria. So that is being attracting that bacteria right to the roots. So you get all the bacteria that starts moving in. And bacteria and fungi as well are basically like little fertilizer bags. You can kind of think of them as that. There's a lot of nitrogen, other nutrients that are stored in those cells, and the plants want it. So they've attracted all the bacteria to them. It's right there. They've got these fertilizer bags hovering around them, right around the roots. Now, how can they get to those nutrients? Well, this is where those biological regulators come in. The nematodes, the protozoa, they're come in as well. They follow the bacteria, they consume the microbes, they consume those bacteria. And when they eat those microbes, they excrete extra plant available nutrients. So they don't use it all up. They use some of it for their own cells, for their own processes within their cells, but they don't use it all up. There's some extra that's left over, and that's left there in the soil. And you know where that is? That's right there around the roots. Okay, so now basically what the plants are able to do is they are able to attract their own little food sources, their own little fertilizer, and then they're using the rest of the soil biology, those biological regulators, to break open the fertilizer and provide it directly to their plant roots. So I just think that's a really cool thing that we see with that, where you have this relationship between the plants and the microbes. Another way that we can see more nutrient availability and increase in that to plants and to our crops is through mycorrhizal fungi. So that symbiotic relationship between these mycorrhizae and the plant roots, the crop roots. So this is a corn crop with, no mycorrhiza right here. So this is the extent of its reach into the soil. The one on the right, we can see it's able to get much further out into the soil because we have this mycorrhiza. So the plant roots are still only right in here, but the mycorrhizal fungi can reach out much further. And so this is really important for phosphorus, because phosphorus is an immobile nutrient. So the plant root can only get, generally, if there's no mycorrhiza, it can only reach the phosphorus that's very, very close to that root right in here. And once it uses it all up, it's out of luck. And that's where you can get some phosphorus deficiency and issues like that, because it can't reach it. But if you have the mycorrhizal fungi here, they are able to attach to that root and spread out from it and reach out much further into the soil and get phosphorus and then send it back to the plant root. And in turn, the plant gives the mycorrhizal fungi some sugars, some food for its work. So basically, they have their own little payment system there, where the mycorrhizal fungi brings in phosphorus. Also really important for water as well, being able to bring in water. And then the mycorrhizal fungi gets paid in turn through those exudates from the plant. So that's another way we can get increased nutrient availability with the soil biology. All right, so moving on now to trafficability. So when we're talking about trafficability, that's the ability to support agricultural traffic without degrading soil. So whether you're out there with your combines, or you are planting or spraying nitrogen or anhydrous, whatever it is that you're doing, you need to be able to get out there in the field as soon as you can, especially after heavy rain events. We want to be able to get out there. And if there's heavy rain events and things that we've seen recently in recent years with the very wet springs, it makes it difficult to get out there because of all of the rain that we're getting. So if there's a way that we can improve the trafficability when we can get out there a little earlier, I think that's a really important benefit from having the soil biology. And how that works is, to get that higher trafficability, we need to reduce the risk of compaction. That's the main issue that we're talking about here. And the way that we do that is by increasing aggregate stability. So aggregate stability, you may have seen this before live, but this is a picture of a slake demo. So this is from Iowa. So this is on soils in Iowa. They have the black soils, the mollisols like we have across central Illinois. And here you can see this is two different soils. They have relatively similar organic matter. But the one on the left has very high aggregate stability. So we can see here, it's holding together. When they put the clod in the water, it's holding together. There's just a small amount that's fallen to the bottom of that cylinder, but the vast majority of it is holding together. Whereas the one on the right, you can see here, is breaking apart into all of these tiny little soil particles and all of these little pieces. And so what happens in your soil, this is what it looks like. So this is a soil that has high aggregate stability. We have the aggregates holding together. We have all these pore spaces in between them. The one on the right, this is a compacted soil. All right, because all of those particles have broken apart and now they're being pushed together. And if you have any kind of heavy traffic, when those particles are all loose like that, driving over those soils, especially if they're still fairly moist, they're going to compact. Okay. And also, we're going to talk a little more about this with the infiltration. The water is still sitting there as well. There's nowhere for that water to go unless it's running off the surface. So you have problems with water ponding, with water just sitting there on there, as well as the fact that these particles are broken apart and they're able to push together and there's nothing holding them to keep those pore spaces in place. So if you have those pore spaces in place and the aggregates are holding together, they're not going to compact even if you have the traffic on it nearly as easily. So how do we get that aggregate stability? How do we get these holding together? And that's soil biology. We need that soil biological activity. So how this works is those aggregates are held together with glues, and those glues are coming from the microbes. So it's a microbial products, chemicals and exudates that they have that are holding those aggregates together. We also have fungal hyphae, which is basically the filaments, the strings coming off of that fungi that are holding those particles together. We also have plant roots. So all of these different aspects of soil biology are really holding those aggregates together. Okay, so here's some more pictures of that. This is one where we have bacteria. Actinomycetes are having filaments that are holding these together. And here we have some fungal mycelia that are doing the same thing, holding those aggregates together. Okay, and so I mentioned this before, we can have issues with the water when we have these low aggregate stability or low stable aggregates. So here we have a soil that has good aggregate formation. And here we have one that has very poor aggregate formation. They are all in separate particles. And so this is what happens to those. So if you have an aggregated soil, we have all these aggregates, the particles are being held together by those glues, and there's all this pore space left, and the water can flow right through. Okay, we also have air that can get through that as well. So here the water can go into your soil. When you have it breaking up into all of these tiny particles, you can get compaction because they're all settling together. You can get soil crusting. And then depending on the topography and the slope that you have of your field, you can also have runoff, or you can have ponding where there's nowhere for that water to go. It's stuck in a low spot in your field and it can't go down because there's no room to go through those pores. And so it's just stuck there ponding. So those are kind of some of the issues that we have when we have low aggregate stability. Okay, so we're going to move on now to some more talking about plant microbe interactions. And so we've talked about this a little bit already. So plants can give off these root exudates, and those can influence the microbes that are living around them. And they can change based on what stresses that plant is under. So we've already talked about the first one, how microbes can increase nutrient availability. But now we're going to focus on the second part of this, which is providing a defense against pathogens. So here we're going to be talking about disease suppressive soil. So there's actually two ways that this suppressiveness works. One is specific. It's focused on a specific type of pathogen, and that's actually what we're seeing in this diagram down below. And what that means is it's just able to suppress a single type, a single species of pathogen. And how it works is basically it has to get vaccinated in a sense. So the pathogen comes in, the plant gets sick, but it's triggering a change in the microbial community. Okay, because that plant, when it gets sick, when it's stressed from the disease, it's sending a signal out to the microbes and saying, hey, I need help, send me help. And so that causes a change in the biome. So now you get a much more diverse microbiome. And then the next time that same plant comes in, that pathogen's still there. It's not completely gone, but because it's there and we've already had this change in the microbial community, it has a memory. It doesn't allow that pathogen to get as much of a hold. It suppresses it. It becomes much more easy for the plant to handle, and it doesn't get as affected. So that's one way that we have it. That's called the specific, for just how a specific pathogen can affect it. But the other is just general. And so that's kind of just where we have starting out. If you have general suppressiveness, that's usually when you're talking about having a very diverse microbial community. So if you have a wide variety of different microbes within your soil, the pathogens can't do as well. They can't get to abundant enough numbers to cause a problem. And that's because they're being outcompeted by all these other organisms. So basically, it's having all these different people, different organisms there doing different aspects of the soil functions, and the pathogens can't seem to take over like we have in a soil microbial community that looks like this, where that pathogen can really take over and start getting to very large numbers and can start causing big problems because there's not enough competition to keep it back. All right. So hopefully through this presentations, I've been able to convince you why soil biology is so important and how they can really do a lot for you in your soils and in your fields. So now we need to talk about how can you recruit them and how do you keep them there? So this all comes back to the principles of soil health, right? So it's pretty straightforward. First of all, we need to protect our soil aggregates. We need to protect our organic matter. Okay. We do that through minimizing disturbance by maximizing soil cover. We don't want to break those aggregates open. We want to keep them in place. So all of those are really important, but actually even more important for that soil biology aspect of it is this side. We need to feed them. We need to fuel them. We need to give them all these different food sources. And we need to do that throughout the year. So maximize continuous living roots. So we want to give them all of those roots because remember, those roots are a food source for a lot of these organisms. And if we don't have living roots out there, that food source is gone. So we want to give them that food source throughout the entire year as much as possible. And we also want to maximize the biodiversity. So we want to get as many different species of plant out there as we can. So we can do that through having an extended or diverse crop rotation. We can do that through having cover crops. And especially if you're doing cover crops, that gives you an opportunity to do mixes that have multiple different species out there. And then you can also do this by incorporating livestock into your system by adding livestock. Maybe they're grazing the cover crop. That's an extra revenue stream. So there's a lot of different ways that we can do that. But the key with that is if you have diverse inputs into the system, you're putting a lot of different food sources in there. That means you're going to attract a diverse soil biological community. So the more diversity you put in, the more diversity you're going to get in that soil biology. And that's really important for all of those soil functions, to have as many different ones in there coming at it from different directions. So that's really the key with that. So that is the end of my presentation. I thank you for taking the time to watch this. I hope you learned about soil biology and what they can do for you and how you can get them and keep them. So thank you very much.